So thanks, Grace. And really, today I'm a mouthpiece for our whole multidisciplinary research community. It's, the slide that was presented to you earlier today with NCI funding for bladder cancer research, where we have a dismal 23rd place among cancers, is true and it's pathetic. But there's a silver lining to this, and that is that because we've had such challenges in getting funding, we've developed what I would argue is probably one of the most collaborative and team science-oriented international groups of investigators, urologists, medical oncologists, and basic scientists. And so the team is built. We've got great technology that we've got at our disposal now. And Rhonda Pino, who just became president of our institution, said it best when he said, this is just a watershed moment for cancer in general and bladder cancer in particular. Everything's coming together right now. We've got the tools to understand everything in great detail, and we've got drugs that are starting to target things in a smart way. It's just everything seems to be coming together, and I expect over the next 5 years you're going to see rapid progress across cancer types, but we hope that we will be active in promoting this in bladder cancer. So from a scientist's perspective, understanding the biology of bladder cancer is going to be critical if we're going to match tumors to the right drugs, which in the end, whether you have non-muscle invasive disease or muscle invasive disease, is the goal. Maybe the right drug is surgery, maybe the right drug is BCG, maybe the right drug for non-muscle invasive cancer hasn't been tried yet. But regardless, the idea is to use the cancer's biology to govern choices about therapy. And so from a basic science point of view, we want to understand how the disease is caused. And as many of you may be familiar, there's a current rage in cancer biology looking at so-called stem cells for cancer. You've heard of stem cells earlier today for generating new bladders. Well, these are kind of similar to those stem cells, except they're the cells that we think might be the cause of initiating the whole cancer. And so if we knew what those cells looked like and we knew how to target them, the idea is we would do a much better job of eradicating cancers, because right now it seems to a lot of us that when we treat patients with MVAC or gemcitabine cisplatin, we may be getting rid of a majority of cells in some tumors. You get complete responses or near complete responses, but the microscopic disease that remains might consist of these stem cells, and they're just going to grow back again. So at Baylor, there's a strong effort to do this, and other institutions across the country are doing this. Also, as pointed out by Seth earlier, we actually have the government's attention in bladder cancer, and we have an opportunity to get all the information possible on a set of 500 muscle-invasive cancers through this Cancer Genome Atlas project. We're all involved in the organization design of that study. We're all actively engaged. We need your help in the patient advocacy capacities that you all serve to make sure all the patients know about this. This is really big stuff. This is the way we're going to really make progress in personalized cancer therapy to get this project finished. It'll catalyze enormous advances in science, as we've seen already in brain cancers and melanomas. It's absolutely essential that we get this done as quickly as possible. We also heard a bit about toxicity and how to predict toxicity in people based on their inherited genetic makeup. There's also a chance that this inherited genetic makeup predisposes some people to cancer more than others. And so there's a strong effort going on across the country to understand how these polymorphisms, these small differences between you and me, might cause you to be more susceptible than I am, or vice versa, to certain types of cancer, or for that matter, heart disease or diabetes. And then finally, the project that I work on the most at MD Anderson is to try to define the extent of heterogeneity. So we think that everybody's cancer is different, but that the cancers fall into subgroups, subcategories, and that understanding which subcategory your cancer fits into can allow us immediately to enrich for therapies that will benefit your cancer and maybe help you avoid therapies that will be absolutely pointless for you to be exposed to. So this heterogeneity is really important, we think, to understand in order to guide both decision-making in terms of prognosis with surgery and so forth, but also in terms of which chemotherapy to select for your particular cancer. So cancer is caused by a variety of genetic mutations, along with changes that are not mutation-based. But one of the major goals of the TCGA effort is to identify all of the mutations that are present in 500 cancers. We've got a head start. We know that all of these genes on this list here are mutated in bladder cancers. And in a couple of these mutations actually have drugs that should target tumors that have them. We have drugs that should affect cancers with this mutation or this mutation better than cancers that don't. And we have examples from other disease types like melanoma, where a mutation in a gene called BRAF causes tumors to be exquisitely sensitive to inhibitors of BRAF, where we have a strong suspicion that knowing whether your tumor has this mutation might allow us to guess that you might benefit from one of these two different classes of drugs. That's something we can do almost immediately to try to advance our efforts to treat bladder cancer more personalized fashion. And then through work that actually came out of a group in China, we've seen a glimpse, our first glimpse, of the genetic mutations across the whole genome in a set of bladder cancers. This paper I'll talk about in just a moment. So what are the implications of knowing that these mutations are present in cancers? Well, I already told you one implication that's very important. Having the mutation might make us able to predict that that particular cancer would be sensitive to a drug that targets that protein, the protein that has that mutation. Another implication is that we might be able to use these mutations to detect cancer, not through blood in the urine, but through tests that measure the mutation in the urine. It might be that seeing these mutations is also a way of understanding cancer biology much better than you would just by seeing cells in the urine or by seeing blood in the urine. And it might be that just in the future, if we can do these kinds of tests on a large scale, that we wouldn't even need biopsy material to really understand what the cancer's biology looked like. You could just do a test on urine, and you get a good picture of what the cancer biology looks like for therapeutic decision making. And these tests are available now. I mean, this is a group in Europe that we're working with to do some mutational testing on one of the trials that was discussed earlier, one of the neoadjuvant trials we're doing. But here's the paper I was talking about, where a group in China actually beat us to the punch a little bit and published a paper just this fall, in September, describing all of the mutations that are present in a set of bladder cancers that they had analyzed in China. And what that means is that we have a platform upon which to build additional analyses and compare mutations in different tumors and understand if the mutations can help us inform clinical decision making and help us subset cancers into various categories. So the mutational analyses are critical. The TCGA effort is critical, and Dr. Lerner has already presented that to you today. I'm going to just echo what he said. Let's talk about heterogeneity a little bit. So we all know that there are essentially two big subtypes of bladder cancers. There are the non-muscle invasive subtype, or the papillary subtype, and the non-papillary or muscle invasive subset. And clinically, they behave very differently, as we've been talking about all morning. But at the genetic level, they're also apples and oranges. They're very different genetically. This pathway tends to be associated with certain panels of mutations, and this pathway with others. And if we turn to our breast cancer colleagues, the way that they've started to appreciate this complexity in even higher detail is they've used some of these whole genome approaches to analyze sets of tumors and ask, okay, do they group into subcategories that are easily understood? So this is a classic paper that was published by Charles Perou's group several years ago, where they suggested there were probably 5 dominant subtypes of human breast cancers. And they did this by using so-called gene expression profiling arrays, which we use in our group too. And they saw that there were these clusters of tumors that fell into these different categories. Now these categories are very well established, and they have important implications for therapeutic decision making. We're collaborating with a group in Sweden, along with Dr. Lerner and others, to do the same thing in bladder cancers. Again, the idea within our community is if somebody's doing it well somewhere else, why don't we just work with that person rather than try to reinvent the wheel? It's leveraging resources. So we've sent our gene expression profiling data on 140 bladder cancers to Dr. Höglund. And this is an example of what he's done with his bladder cancer cohort. So these guys over here are non-muscle invasive, and these guys over here are muscle invasive. And already you can see that he thinks there are two subsets of non-muscle invasive tumors that he can subclassify very easily, and up to 5 different subsets of muscle invasive cancers. It would make a big difference to know which type of cancer we're dealing with in terms of therapeutic decision making and prognosis. We want to do something about all this molecular profiling and genetics immediately. We want to have immediate impact. Long term, we want to find drugs that target tumors that have specific spectra of mutations. So if you have FGFR and PIK3CA, we're going to have a cocktail of drugs that we would offer you to help kill your tumor. But immediately, we need to do something, and the way we can improve things immediately is to try to understand where to use cisplatin-based chemotherapy. How do we use it? How do we identify the tumors that are going to respond to this, to the patients that are going to benefit from this? And then on the piggyback of that effort, to do the same thing as each new drug comes along, to try to determine whether there's a way of predicting which tumor is going to respond to it by mutation or other strategy, and then do a multi-center test, do a clinical trial with a lot of different groups across the country to test the hypothesis that the presence of this mutation and the presence of this gene signature is going to predict responsiveness to this therapy. So we organized a big collaborative workshop again at the National Cancer Institute this past September. And as part of the science, we talked about a lot of different things. We talked about stem cells, which we've already talked about. We talked about trying to find cancer cells in the peripheral blood. So you get a blood test and you find cancer cells. And what does that mean in terms of prognosis? And can we measure the biology within those cancer cells and tell something about how to treat your tumor? Again, we talked about mutations and which ones might be druggable right now, like FGFR3 and PIK3CA. Whole genome analyses, Dean Bajorian talked about this hereditary component, the factors in the background of your DNA that might make you more susceptible to bladder cancer or respond better or worse to certain drugs, or maybe experience more toxicities to certain drugs because of their metabolism in you. We talked about statistical methods for analyzing these large data sets. If you sequence a whole genome, it's terabytes of information that get parked in a server somewhere, and we're trying to figure out how to streamline that process so that you can actually do this on every patient who walks in a cancer center's door. And then finally, we wanted to generate a product. We wanted to generate a clinical trial that would test a biology-based hypothesis. And the trial we discussed is one that Dr. Lerner's been actively involved with as well. It's going to be run through the Southwest Oncology Group, and it's going to test the predictive benefit of gene expression in predicting cisplatin-based chemotherapy efficacy. And this is a slide from Dan Theodorescu, who's the leader of this effort. He's developed a way of sampling cancer biology using whole genome expression profiling. And using that information, he's been able to develop a computer algorithm that he thinks will predict whether you'll respond to cisplatin-based chemotherapy. And right now, as you've seen all morning, this is what we're dealing with, where everybody gets GC or MVAC. It's the standard of care. And this is the outcome you get with either one. But embedded within this, there are obviously going to be some people who are doing really, really well because of the chemotherapy. And using Dan's Coxon algorithm, what we want to be able to do is this. So if you are one of these people, you would get cisplatin-based chemotherapy. If you're one of these people, you get something else. You'd avoid the cisplatin-based chemotherapy altogether because it's not going to benefit you, and it's a waste of time. You want to spend your time wisely. You want to get right to the things that work as quickly as possible. Why would you go on months on a regimen that's not going to be helpful to you? This is the way it works. You don't have to go through the details. You've got it in your handout. They'll be on the website. Bottom line is just like us, he looks at the whole genome, in this case at the RNA level, uses his computer to predict whether or not your tumor is going to respond or not to cisplatin-based chemotherapy. And the idea of this clinical trial is to test that hypothesis. What's interesting is that in this case, what Dan was doing and what we were doing converged. It's just the way science works. If you're both working on something and you're probably on the right track, oftentimes you meet together at a point downstream. We were looking for ways of predicting chemotherapy sensitivity in bladder cancer cells, and we discovered something called epithelial to mesenchymal transition that seemed to be predicting platinum sensitivity in our cells. And when we compared our way of looking at things to his way of looking at things, they overlap perfectly. So Dan, using Coxon, identified the same chemotherapy-sensitive cells that we did using our EMT approach. So we're going to be testing a lot of different hypotheses under the umbrella of Dan and Tom Flake's clinical trial within SWOG, generate a lot of information, and quite frankly, we all hope that this is going to tell us that we can actually do personalized medicine right now with conventional chemotherapy. And if this proof of principle turns out to be a success, this is the platform we would use to introduce new drugs in bladder cancer patients, one after the other, systematically. And what's interesting is that when we look at other novel agents in this space, we can see non-overlapping effects. So some cells are sensitive to platinum and insensitive to other things, whereas different cells can be sensitive to these other things and totally resistant to platinum. And this is one of these cases. We've got a drug we've worked on that's a little like Taxol that actually seems to kill the platinum-resistant cells in a non-overlapping way. And so if this could be harnessed to set up a clinical trial, for instance, where you put some patients on platinum based on their gene signature, other patients on a drug like this one, you might actually get to the point where you're not just benefiting the 20% or so of patients that you started with platinum-based chemotherapy, but maybe we can kick that up quickly to 40%. So the other thing is prognostically, having this information is really important. We've already been able to subset our cancers based on this marker and see right up front, just based on a molecular signature. We talked about grade earlier and stage. This would be a different way of looking at cancers, using molecular markers to define grade and stage. We already know that this one's really important. It has a major impact on disease-free and overall survival in muscle-invasive cancer. So embedding molecular markers into the diagnostic and prognostic aspects of cancer management is going to make a major impact. And maybe it's someday having a set of molecular tests will replace conventional pathology. Rather than having different stages or different grades, we have a molecular test that tells us exactly what the biology of that tumor looks like, why it's dependent on the things that it's dependent on, what it's sensitive to, and how lethal it might be. So I want to leave some time for questions. This is why I kind of blew through a couple of the slides there. But the bottom line is that we're entering a brave new world where this kind of high-throughput, data-dense analysis of cancers is going to enter the mainstream. And it's already entered the mainstream in other disease sites like melanoma and brain that we've talked about. We want it to enter the mainstream even more quickly in bladder cancer. We've got a nice, strong, multidisciplinary group assembled. We have a head start with regard to the mutational analyses that have already been performed in the 60 tumors we're into with TCGA. We want to be as efficient as possible in getting these discoveries into clinical management of bladder cancer, and we need your help. We need you to be engaged, and we need you to be out there telling everybody you can think of how important this is to you. The more cooperation, collaboration we have from everybody, including you, the more efficient this whole process is going to work. And again, I think the trial that I talked about through SWOG, the Coxon trial, is going to open soon. This is going to be our first step into personalized medicine. It's just the first one. We've got many steps after that, but it's going to happen very soon. This is a brave new world of personalized cancer medicine that we're entering. So this is the key: clinical trials, surgical trials, therapeutic trials. We want to get these answers as quickly as possible, and the clinical trials are the best way of doing that. So these are just some of the key contributors, collaborative contributors who are contributing to all of this effort. We have the only spore in the country, which is a tragic situation. Prostate has a lot more. But fortunately, this spore has helped, along with other cooperations, in assembling teams of investigators that go beyond MD Anderson or Houston to attack some of these questions. And so, again, the idea that we're on the verge of making real progress in this disease site is real, and we're attacking it as a team, and we need your help. So I'd be happy to take any questions you might have. I think, do we have to break right at 1:30? No. Okay.